Hello everyone, in today's video I will show you how to perform cross-docking step by step. And trust me, this method will make your docking study much stronger and more publishable. We will use three crystal structures of base 1 whose PDB IDs are 2VA7, 2VA6 and 2VA5. And I will guide you from downloading files all the way to RMSD analysis. So let's get started. First, let me explain quickly what is the difference between redocking and cross-docking. In redocking, we dock a ligand back into its own protein structure. But in cross-docking, we take a ligand from one structure and dock it into another structure of the same protein whose conformation is different from the one whose ligand we are using. This helps us check is our docking protocol reliable. Moreover, can it handle the protein flexibility or not? That's why cross-docking is considered more realistic in drug discovery. First, go to Chrome and write PDB and then click on the first link, which is the Protein Data Bank website. Here, download the three base 1 structures that are 2V, A7, 6 and 5. Click on it and then from here download the PDB format of this structure and then click on the save button. In the same way, download the other two structures that are 2VA5 and here you can see and 2VA6. Here you can see. Download these two structures. Each of these contains a co-crystallized ligand which we will use later. Now open the downloaded structures in PyMol or Discovery Studio for refinement purposes. In this video, I will use PyMol software. Open it, click on the file and open one of the co-crystallized complex. Here you can see that this green color represents the protein. Moreover, these pink color dots represent ions. There are 3 ions and this C27 is a small molecule and these dots represent water molecule. So to refine the protein, you have to remove these ions, also remove the water molecule. Now click on these ions. This is the selection. Click on the selection and action button. Click on remove atoms. It just removes the atoms. Now it's only the protein sequence and here is a small molecule. Click on the small molecule and just extract. You can copy to another object or extract the object. It's totally up to you. I'm just clicking on extract. It just give it another name, object 1. Here you can see that this is the protein 2VA7 with chain A. Moreover, the extracted small molecule is here. Now just deselect this selection and object and click on the file to save this refined protein. Moreover, you can add the polar hydrogen. Otherwise, you can all skip and add these polar hydrogens in PyRex software. Now simply save this protein structure, export molecule and just click on the save button. Here click on the PDB extension and write at 2VA7 refined structure and just click on the save button. It automatically saves the protein structure. Then click on this object, small molecule and click on the export structure. Save this small molecule in the same way in PDB format. You can also save it in mol2 format, STF format. I am going to save it in PDB format. It's totally up to you in which format you are going to save your small molecule. I will name it 2VA7 molecule 1 or ligand 1. You can also name it ligand 1 molecule 1, which name you want to give it, and simply save this file. In this way you can extract your small molecule and refine the small protein structure too. Do it with the other two co-crystallized complex. Here you can see that I refine all three proteins and extract the three molecules with these names. Now for docking purposes, open the Pyrex software and simply import the three proteins and ligands. Then we will convert the ligands into PDB format after minimizing the energy. From here, you can load the protein structure. Okay, open the structure one by one and click on it, make it macromolecule. When it makes your protein as a macromolecule, it simply add the polar hydrogens and the other energies. Do the same with other two structures. Now import the small molecules. Click on this open babel and click on this option. Select the small molecules, molecule 1, then one by one open the other two molecules. And now molecule 3. Right click and click on minimize all. It simply minimizes the energy. Then right click and convert all to autodock ligand. It simply convert the PDB files into PDB file format, which is required for docking. Then click on the Vena wizard and click on the start button to start the docking. Now set the grid size. For this, you have to use the co-crystallized ligand position to define the grid box size. The grid center should be the ligand center and the grid size should cover the full binding pocket. For this, you can use the original protein structure with the small molecule. I just open this one and now I will highlight the small molecule which was C27. Here you can see at the end of the protein, it's here. Now I just label for display purpose. And here you can see it just label the small molecule. Okay. Now I have to set the grid around this co-crystallized ligand and you can see the grid box. You have to move a bit here and now yes, it's alright. Now I just deselect the original structure and minimize it. Now I perform the cross-docking. Here I use the structure 2VA7. Now I will select the small molecules, molecule number 2 and 3, which is from 2VA5 and 2VA6, not the molecule 1 which is extracted from the 2VA7 complex. Just click on the forward button. It just starts the docking. You don't have to dock the ligand into its original protein. Like I will not dock the molecule 1 with 2VA7 because it extracted from the 2VA7 complex. It just takes some time. You have to check the binding energies and visualize the binding pockets. Now compare how well the ligand fit in the different protein structures. Different protein structures mean different conformations of the same protein structure. Now perform the docking of 2Va6 with molecule 1 and 3. Then perform the docking of 2Va5 with molecule 1 and 2. And then we will visualize via PyMol software. You can save the file in CSV from here with all the binding energies and RMSD values. Just save the file. Select and download the best doc post by right clicking on the doc post and just click on the save as PDB. It saves the doc post into PDB file format on the location where you want to save the file. After that, you have to calculate the RMSD values. For this, firstly we have to make the complex file of the doc post. For this, we will open these two poses and the refine 2Va5 structure. Now select the molecule 1 with dock pose and the refined protein structure and click on the file and export structure into PDB file format. It just makes the complex of the docked pose because in PyMol you have to compare or align the docked and the original native complexes, not the small molecules. In the same way, you have to do with the second molecule. Export molecule and save it in the PDB file format. Okay. Firstly, we save only the dock pose. Now we save the complex of the docked pose. Just save this and now delete everything. Now we will compare the docked poses with the native conformation. So for this, open these complexes. Open the first complex and compare with the native structure. Now click on the align to the molecule 2Va7. It just gives you the RMSD value of 0.17. Now the same way we have to calculate the RMSD value for the second molecule. For this, delete everything and then open the second molecule. And then click on 2Va6 because second molecule's native structure is in the 2Va6 file. Then align it to molecule 2Va6 and it provided the RMSD value of 0.266. If the RMSD values are less than 1, it considered excellent, while the RMSD values within the range of 1 to 2 is acceptable. Moreover, the RMSD values above the 2 angstrom considered poor. So, if you have the lower RMSD value, it means there is better docking accuracy. Moreover, don't rely only on numbers. Also check the hydrogen bonds, check orientation and overlap of ligands. If RMSD is low across structures, it means your docking protocol is reliable, your results are not biased, and your study becomes stronger scientifically. If you learned something new, like this video, subscribe for more bioinformatics tutorials and share with your research friends. In the next video, we can cover how to write these results in your article. If you want this video, you can comment and I will make the video on it. See you in the next video.